Hello everyone, thanks very much for coming. It's great to see such a good turnout. Gives me a lot of pleasure to introduce Kristen. She's one of the rare people that walk through your door with a fully formed project and funding to go with it. So, and she's continued to work in a very independent and productive way. She's continued to gain collaborators. She's worked really well with people and funders to keep the funding going for her project, and in her time as a PhD student has even built her own laboratory. So I feel like she's been a wonderful student, and she's produced some really nice work, and I'm pleased that she can present that to you today. Thank you.
Okay, so today I'm talking about my PhD research, which is on conservation of rare and enigmatic fauna using the smoky mouse as a model species. I would like to first acknowledge the traditional owners and custodians of the land in which we meet today, the Ngunnawal people, and pay my respects to their elders past and present, as well as to emerging leaders. And I extend that respect to all Aboriginal and Torres Strait Islander people. Sorry. It's not working now?
I think it's just because I could try again.
Okay, so acknowledgement to country. Okay, sorry. Okay, so it's no surprise to everyone here today that the world is facing a biodiversity crisis. Modern extinction rates are hundreds to thousands of times greater than baseline extinction rates. Conservation action is needed more than ever. However, this requires a sound understanding of species-specific knowledge. Small and declining species are especially difficult to study in the wild, and the population estimates are often unknown. In Australia, small species also often suffer from what's known as niche reduction. So the niche is the ecological role that an organism plays within its environment. I have a plot here that's illustrating the different facets of a niche, both with threats present and where there's severe impact, and where there's threats present but that impact is reduced. So the fundamental niche, which is the entire breadth of conditions in which a species can persist in, is illustrated in the light green shaded area. And then the historical niche, represented in the dark shaded area, represents the area that the breadth of the conditions the species lived in, or species could persist in before European colonization. And then we have the contemporary niche, which is showing how those species are existing with the threats of European colonization. So this includes habitat fragmentation and destruction, and cat and fox predation. So essentially this is showing us that the conditions in which they live in has been severely reduced since European colonization. And a good example of this is grassland species, where they're highly vulnerable to predation by cats and foxes, and many of them have sought refuge in more denser habitats. So because of this, our ecological knowledge of these species is inherently limited. These species also benefit from both in situ and ex situ conservation strategies. In situ is within the natural range of the species, so managing populations within the habitat, threat controls. And ex situ conservation is anything outside their natural range. And so that's largely conservation, breeding or captive breeding programs is a big part of that. And it's especially important to consider both these strategies. It's especially important to consider conservation translocations and their role in both these strategies. So conservation translocation is the intentional movement of an organism or a group of organisms from one place to another for conservation benefit. And we can use this to supplement existing populations where their numbers are low and where threats are controlled, or we can trial translocations into new areas where threats are controlled. And a good example of this is translocations into predator-proof reserves. Okay, so this is a very broad overview of how we go about conservation with some examples given in each sort of state. So we start with the status of the species and the populations. Typically, we start with conducting surveys. So we want to know a bit about their range, the genetic structure and their diversity. And then we need to understand a bit about their population dynamics, so population trends and gene flow. And then we need to make sure we have a good understanding of their threats. So these include environmental threats, such as habitat destruction, depletion of resources, predation, competition, and disease. And then demographic and genetic threats, which can include inbreeding, stochasticity, sex ratio, and age structure. And then we move on to the recovery actions, and these can be either in situ, so habitat restoration, threat control, population management, or ex situ, predominantly captive breeding, which is what I'm going to be talking about today, but also things like biobanking, which is like the storing and preserving of biological samples, like semen or eggs. So now I'm going to tell you just a little bit about my study species, the smoky mouse. So some of the things that we do know about the species. They're critically endangered nationally, endangered nationally and critically endangered in New South Wales. They have a low reproductive rate, so they breed seasonally. They'll have 1 to 2 litters per year with 2 to 4 pups per litter usually. They're omnivorous, so they have a diverse diet ranging from hypergel fungi to native seeds and plant and flower material. They were first described in Victoria in 1934 and were since recorded in 5 geographical regions, but there have been no detections in ACT, the Otways or Gippsland since 1988. And so this map here shows the detections and the predicted habitat using the bioclim climate model, and this was I pulled out from the. Oh, sorry. Sorry, threats. Yeah. So this map here shows the detections and predicted habitat using the bioclim climate model. The dark grey area represents the core predicted habitat, and then the light grey represents the marginal habitat. And this is just how up until now, we've been sort of grouping the species into these 5 geographical regions. So we have the southeastern highlands, which is like the central and eastern Alps of Victoria, as well as Kosciuszko National Park and ACT. And then we have the Grampians out to the west and the Eden hinterland or the Nulka population on the east. And then East Gippsland and the Otway range are the populations that have since gone extinct. Okay, so some things that we don't know. We don't know a lot about their population connectivity, their sizes and their demographic trends. The cause of range decline and conditions required for recovery are not well understood. And we don't have a great deal of information about their general ecology, so their behaviour, sociality, breeding systems and habitat requirements. Okay, so the overall objective of my research is to address key knowledge gaps to guide conservation strategy for the smoky mouse. And I'm going to follow this workflow that I presented before, so the status threats recovery workflow. So first I investigated population connectivity, gene flow, genetic diversity and demographic trends. I then leveraged that genomic data to reveal demographic and genetic threats to the species. I then applied both experimental and adaptive management approaches to advance captive breeding and translocation recovery strategies. Okay, so starting with status. We know that they've experienced range declines, especially since European colonisation. How do we group these species into management units, also termed MUs, and what is their genetic diversity? In question 2, what I'm going to be asking is what is the demographic history and is there gene flow? Okay, so for this analysis, I used tissue samples that were collected across the species model range between 2010 and 2021. And it was really important to exploit all available genomic data so that I could maximize sample sizes and range coverage across the species' geographic distribution. So to do this, we had to actually incorporate 2 different sequence methods. So I had 48 tissue samples that we sent to DART for sequencing to generate SNP data. And then there were also whole genome sequences from 52 individuals that were owned by Museums Victoria for a different study. Museums Victoria. So we had to integrate those 2 data sets so that I could really make use of all the information we have. So to do this, we had to first align the whole genome sequence data and the DART SNPs to a high quality reference genome. We use the broad-tooth rat because it's a similar species. And we can then call specific SNPs from the whole genome sequences based on their DART SNP location in reference to that genome. So we then pull all those SNPs together into a single data set and performed bioinformatic filtering for downstream analysis. Okay, so these are the first results from that data. So the top left, we're looking at a PCA plot, which shows the genetic distance between genetic clusters. So we can see that there's 5 distinct clusters here. We've got Kosciuszko National Park, the Grampians, Central Victorian Alps, and the Eastern Victorian Alps and the Nulka. So Nullica and Eastern Vic Alps look like quite closely clustered there together, but when I run another plot and take out the Grampians, they pull apart quite nicely. So it's just because we've got so many populations there and they're all so different, the clustering is a bit tricky. I then ran a genetic structure analysis using Pritchard's software, and that's what you're seeing on the right. So these pie charts are color-coded according to the groups that have been assigned based on the results of genetic clustering through that analysis. And the first thing you're going to notice is that like most of them are just all the same color, and what that shows is that there's very distinct clustering for all those regions. We do have signals for admixture in EVA, that's the Eastern Victorian Alps down there, so you can see how that's made up of some of the compositions of Nullica, KMP and CVA, but that was only a very small sample size. We only had 3 individuals for that, so that might also be skewing the results a little bit there. And then looking back to this map that I showed you earlier, where we had a kind of predicted range joining those Southern Highland populations, we can see that it hasn't really taken place in these results. We can't really see that well through the results. We have like really distinct clustering for CVA and Kosciuszko National Park. And then in the center is where sits EVA. So that could actually be signal for like a real signal for admixture. Maybe that was once a connected population. But we're not detecting it at either edges of that range. So if there is gene flow, it's very little, or maybe it's only one way going into EVA. Okay, so then looking at genetic diversity. So the first plot here is expected heterozygosity according to each population. And we can see that the Grampians has relatively low heterozygosity compared to the other populations. And then on the right, we have number of private alleles. And this is a measure for genetic uniqueness within each of these. Sorry, can you keep just reminding me to do that? So on the left here, we have genetic diversity measures. We have expected heterozygosity against each population, and the Grampians has relatively low genetic diversity. And then on the right, we have the number of private alleles for each of these populations. And that's a measure of genetic uniqueness. So even though Grampians have low genetic diversity, it still has quite a high number of private alleles. So there is unique diversity within that population. And EVA is really small. We've only got 5 private alleles in that population. Again, it's a very small population. So we probably need to be expanding on that sample size a bit to get a more accurate reading. But it's also not something, it's something that we would expect to see if there was a high degree of admixture in the population as well. Then I looked at demographic history for the species. So I used the stairway plot 2 method, which uses non-parametric modelling to infer changes in effective population size over time. Effective population size is like the measure of an idealized population. So it's the number of individuals that would effectively contribute genetically to succeeding generations, and it's often smaller than census size. I also excluded EVA from these analyses because of their small numbers, the small population number. So on the left, we have, these are all plots showing the trend in decline over time. The plots on the left are showing a period between current day and 10,000 years ago, and the plots on the right are showing a period between 100 to 100,000 years ago. And then we've got horizontally across each population. So we can see that there's a gradual decline, trend of decline over time, and that there are some like stand-up moments. So between 20 and 50,000 years ago, we can see that that decline really starts to take off for all populations. During this time, regular burning started across Australia. We also had megafauna extinction and the last glacial maximum. So there were a lot of changes to landscape, vegetation structure, as well as trophic structure during that time. And then 250 years ago, we see a very steep decline. And this, of course, coincides with European colonization. It's important to point out that this method's good for inferring changes over time through a long period, but it's not very good at obtaining accurate estimates. So we're not really looking at current day estimates from this, but we'll get to that a bit later. So this is just a zoomed in view of the present day to 1,000 years ago, which again shows that sharp decline for all populations, and the graded area is 95% confidence interval. And we can see that they've fallen below 1,000 between current day and 250 years ago. And an effective population size of 1,000 is recommended for long-term population viability. So that's quite significant. Okay, so how do we group into MUs and what is their genetic diversity? We have 5 major clearly distinct MUs. Grampians have very low diversity, and all populations except for EBA had plenty of unique alleles. 2, what is the demographic history and is there gene flow? There's a trend of decline over time, especially since European colonization, and possibly one way for gene flow. Otherwise, there is no evidence of gene flow in any of the other populations. Okay, moving on to threats. So we have a decent understanding of the environmental threats, at least where they are currently. The question that I'm asking here is the current degree of isolation and inbreeding a threat to population persistence. So we're focusing on the demographic and genetic threats here. Okay, so here is inbreeding coefficient against each population. And again, we can see Grampians is a standout. Grampians had a really high inbreeding coefficient compared to other populations. It is important to know that an inbreeding coefficient of greater than 10%, so greater than 0.1, can result in reduced fitness. And then we have the contemporary effective population size. And I did this using the linkage disequilibrium method, or the LD method, which is much more suitable for contemporary estimates. And it's actually quite hard to see because they're so small, but this is a bar plot. There are bars there. So the highest was with Kosciuszko National Park at 32.5, and then we went right down to 3 for Nalika and 2.1 for the Grampians. So they're incredibly low. And any of less than 100, and any of less than 100 demographic and genetic factors interact to generate what's known as an extinction vortex. So these are very critically low numbers. Okay, so is the current degree of isolation and inbreeding a threat to persistence? Yes. Any was critically low. There's little to no gene flow. All populations are highly vulnerable to the effects of inbreeding and stochasticity, so they're at a high extinction risk. Okay, now moving on to recovery. So we do have measures in place for the protection of critical smoky mouse habitat at the current ranges, as well as cat and fox culling, especially in the Nalika area. For ex situ strategies, we have yet to develop effective translocation techniques for the species, and they're also very difficult to breed in captivity. So there have been a few breeding programs over the last couple of decades, and they generally report highly variable outcomes that are also quite poor. So you'll have 1 or 2 females that will produce quite a lot of offspring, and then most will produce few or no offspring. So the questions I'm asking are, how do we maximize breeding success and population viability in captivity? And what are effective translocation techniques for the species? Okay, so breeding programs are really resource intensive. I've got a few examples up here just to show you. So we have the eastern barred bandicoot, which is quite a success story. They've been running breeding programs for about 20 years for that species. Then we have the numbat. They've been going for about 30 years in captive breeding efforts. The California condor has also been going for 30 years, and it's cost $38 million US so far to translocate 177 individuals into the wild. So very expensive. And then we have the crimbit tinker frog, which took a team of scientists 12 years to get the first hatchlings from. So they're really resource intensive. They take a lot of time and cost a lot of money. So it's really important to improve breeding success or maximize breeding success, not just for productivity and resource use, but because it's key to animal welfare and viability as well. So the conditions that are good to breeding success are also good for animal health and well-being in general. So if you have happier animals that are performing better, they're going to be healthier and perform better in the wild as well. So there's a range of factors that influence reproduction, and these can be biological, including age, genetics, and condition or diet. And then we also have environmental factors. So we have things like breeding cues and light cycling and heat. And then we have stress from inappropriate conditions. So stress is often overlooked in conservation breeding programs, or it's what's the word? It's superficially considered. So there's often not many analyses gone in towards it. But stress is really important because it affects breeding success through multiple avenues. It affects psychology, physiology, and behavior, which all then go on to affect reproduction. Okay, so now I'm looking at what factors have been affecting breeding success for the smoky mouse in captivity. So early on when I started this PhD, I observed some atypical behaviours for females that were housed on enclosures on the bottom row of a 3-tier enclosure rack. These behaviours included, they were active during the sleep period, they were flipping food bowls, tossing bedding, and we also had a loss of litters for those females. And then when I plotted that out using previous year's breeding data, we can see there's a trend here. So Number of offspring weaned is the mean number of offspring weaned per breeding female per breeding season. And then we have the enclosure rows down the bottom. So there seems to be a trend where the lower down, more lower down the animals are housed, the poorer the breeding success is. But the problem is that this doesn't make sense. There's actually no mechanism there, so we're missing something. So then I took a bit more of a broader look at what was going on. So I collated all the husbandry data that we had. And I ran multiple linear regressions to test various variables and their effect on breeding success, or the relationship with breeding success. So I had biological factors such as age, inbreeding coefficient, generation time, year that season was running, individual ID, and diet. And then I also had environmental factors, which I coined disturbances, because they were based on things that could be giving like a negative influence to breeding success. And you can see where these were located on the illustration I've got here. This is like a floor plan layout of the enclosures. And so conditions varied based on where the enclosures were positioned within the facility. So I looked at conditions like sunlight, proximity to entrance door, proximity to the food prep area, view of husbandry staff, and then the cumulative number of disturbances. So how many of these disturbances each enclosure was exposed to. Okay, and the best fitting model showed that the number of disturbances explained the variation in the data best. So number of offspring weaned per breeding female per season had a strong relationship with the number of disturbances. The more disturbances we had, the poorer those animals bred in captivity. And this also explains the initial observation of that trend with animals housed on the lower enclosures breeding more poorly, because enclosures that were positioned further down had more exposure to these environmental stresses. Okay, so this kind of makes sense now. Enclosure position low, increased exposure to environmental stresses, and reduced breeding success. And we can draw a hypothesis from this, that minimizing environmental stresses will improve breeding success in captivity. Okay, so to do this, we actually built a whole new facility. So we made a couple of changes in the short term at the facility, and there were changes that we wanted to make that we couldn't do at that facility. So this is the plan that we came up with, the layout that I came up with. And essentially I followed a 4-step framework to address the key things that are instigating stress in captivity. So first I looked at functional design. So things like human traffic, noise, vibrations. So that was really important for planning the layout. So we had the food prep separate to where the enclosures were housed. I had really well insulated walls. We had vibration reducing matting in the footing for enclosures, and just generally keeping a bit of quiet in there. And then we had to increase enclosure space and provide opportunity for the animals. So this is mainly ensuring retreat space and ensuring animals can self-regulate their behaviour, and also encouraging natural behaviours. So I used really big enclosures, and I had different zones in each of these enclosures and provided a lot of different retreat spaces, so different hides, nest boxes, different shaded areas, so that they could self-regulate their exposure to external stresses. I also provided areas of natural substrate so that they could be digging burrows, which they would naturally do in the wild. Then optimized climate. So these were things like temperature, light cycling and light intensity, and air quality. And then minimized interference. So it was a very hands-off during the breeding season, not touching animals. Everything was observed remotely. So I came up with a design for an invisible infrared nest cam that sits at the back of the nest box, so that we can perform all our welfare checks remotely. And then also incorporating adaptive management into that. So when we're observing something, keeping track of those behaviours, and then we can record what happens as a result, and we know when to intervene and when to leave them alone. So addressing each of these is a little more complicated than just like a 4-step procedure here, and it's really species dependent, so it will vary on your species. But I found all the resources from laboratory studies on rodents was actually really useful in this, because it all happens in controlled settings. So looking at what the specific effects are, so how light can affect breeding, how noise can affect breeding, that's a really good source for actually understanding those mechanisms there. And this helped me to kind of design, and also it helps because they're closely related to the smoky mouse with a few differences. But it really helped me to design these conditions. So lighting, for example, we know we have to keep seasonal variations if they're seasonal breeders, which we do. We know that they can't be too intense. So working out what levels are too intense and what aren't intense, that's a really good place to start in the literature. And then also other things like light wavelength, which is probably overlooked a lot of the time. So animals have different eye structures to like we do. Mice have a very different composition of rods and cones in the eye. They see light very differently. There are colours they don't see that we see, and there are colours and vice versa. So that mice tend to see everything like really well in the ultraviolet spectrums, and they don't see so well in the red spectrums. So when we're using light, we need to make sure that we're using light that they can see, so ultraviolet wavelength. So just going to the store and buying like a warm light is probably not going to give you the same effect that you want if you're trying to simulate natural conditions. Okay, and so breeding success improved under the new conditions. So I've got a plot here showing the mean number of offspring weaned per breeding female per season at the old facility and at the new facility. And the sections I've got in green are where we implemented some sort of within-house changes to minimize stress. So we provided extra nesting material, and we really worked hard at decreasing the amount of disturbances in those enclosures. In 2021 for the old facility, though, there was a big change around. They had to move things. They were bringing up new rooms and things. So there was a lot of disturbance going on there. So that kind of explains the drop there. And then, so we've improved breeding success, and what's really great is that it's been consistent. It's only been 2 years, and we're actually looking like we're probably going to do better this year as well, which is really good. And we also gained valuable information on the husbandry, breeding preferences, sociality, and behaviour for these animals. Okay, so now looking at maintaining genetic viability in the captive colony. So with the genetic tools that we have, we can monitor rate of loss of diversity in the captive colony, so we can inform when we need founder selection. And we can also use genetic-based pedigree tools to manage the breeding of pairs to maximise genetic diversity. So this is an example of what that looks like on the left. This is a pairwise kinship table showing the pairwise kinship values between individuals in the colony. We've got males on the y-axis and females on the x-axis. So you can actually go ahead and design like your breeding pairs based on maintaining the lowest pairwise kinship values. We can also use things like strategic genetic founder selection. So when we're wanting to supplement the colony, knowing where to source those animals from is really important. So we're really targeting areas where there's high diversity and high number of alleles. And it's also really important to have this data for informing any future genetic rescue strategies. So I'm not saying that we should be doing genetic rescue, and I'm not trying to pitch that and say why it works, but I'm saying that we have the tools here and we're able to do that. We can run simulations to trial crosses, and we can also trial crossing those individuals in captivity. Okay, so next I looked at the trial translocations into Tippimbilla Nature Reserve. So Tippimbilla is a 17-hectare predator-proof reserve in ACT, and a lot of that habitat corresponds to some of the habitat where the species used to be found in the ACT, being the Namadgi National Park. So I used soft release techniques, so short-term containment for up to 4 weeks, and provision of burrows and supplementary feeding. And this was to acclimate the animals and also to get a bit of site fidelity. So we wanted them to kind of hang around as much as they could. A couple of the biggest causes for failures in soft releases are mortality, and then also hyperdispersal, where you release animals and they will just run off right away and you never see them again. I also incorporated non-invasive monitoring techniques. So I adapted a low-cost ring scanner that was suitable to this site, which was really handy. So the animals were microchipped, and we put those around the entrances to the burrows and the feed stations, and we could detect when they were coming in and out. So one of those units was about $180. Versus the Trojan ones, which is the kind of big brand one, which costs thousands of dollars per kind of site you're doing. So it's a big difference in price. And we were able to detect the animals for up to 8 weeks, but then they did eventually disperse. And once they did that, we had trouble finding them again. So that's a big problem that we're facing, long-term detection. We do have partners in New South Wales that are trialling radio tracking for the next release they're doing down in Nagata. So that'll be really exciting to see if they can get the collars working. I had avoided that because there can be a lot of welfare implications with that, especially with small rodent species. So it's a very tricky thing to try and perfect. We also identified new threats there. So I had Bronte Tozer as an honours student taking on an honours project here with me. And she was looking at the overlapping activity patterns between black rats and smoky mice. So when we conducted the initial release, we noticed that the smoky mice started, they kind of stopped showing up when the black rats started showing up. So there was kind of like overlap that when the black rats came, the smoky mice stopped using the resources. And so she actually did her honours project on that, and she did determine they do have a very significant niche overlap, and that the occurrence of smoky mice was significantly decreased when the occurrence of black rats started. We tested some adaptive management strategies to try and reduce this impact. So this was predominantly a whole size reduction. So we measured that we identified what the critical smoky mouse body whole size was, which was 29 millimeters, and then we used that on the feed stations and soft release enclosures, and the rats couldn't access that. And Bronte found that that did deter rat activity, decreased rat activity at the release sites. However, they were still there in small numbers, and this may have still been enough to kind of deter the smoky mice, because we still saw that similar pattern. Even though they were coming far less frequently, when they did start showing up, the smoky mice were still leaving. And that's just a picture there of a black rat actually predating on a smoky mouse. So that was very sad, but it also reveals that they're not only a competition threat, but they're also a predation threat to the species. So very important when you're planning translocations to note that. So how do we maximize breeding success and population viability in captivity? We refine husbandry practices, minimize stress, and use strategic genetic management. What are effective translocation techniques for the species? So soft release for site fidelity worked short term, so we managed to keep them for up to 8 weeks after release, but they do eventually disperse. And it has also revealed that there's potentially threats still present, even though the cats and foxes are absent, and this being the black rats. Ring scanners and cameras were also very useful for short-term detection, but again, we need something a bit different for the longer-term detection. Okay, so conclusions. Identified fragmentation, isolation, demographic and genetic declines, indicating that the species is highly vulnerable to extinction. We saw steep declines since European colonisation, and this also precedes habitat surveys. So just back to that reduced niche hypothesis, that what we're actually seeing the animals in now is a limited subset of their former broader fundamental niche. So we may actually be missing out key information on their habitat use and habitat requirements based on their current range. It also suggests the need for genetic rescue or demographic increases, which again, I'm not pitching through here, but it's something that needs to be considered. More broadly, it represents a practical approach for informing on population status and trends where census data is missing, so very important for small, highly elusive species. 2, captive breeding success improved following an experimental framework and a logical course of action to minimise stress in captivity. This is really crucial to program sustainability, animal health, welfare and viability. And more broadly, stress is a big issue, and it's really superficially appreciated in conservation breeding programs, but the methods I've used here are quite broadly applicable. And third, I trialled translocation techniques into the predator-proof reserve, which gave us really important information for planning and informing on future translocations. We found that soft release provisions encourage short-term site fidelity, and that removal of the perceived primary threat may not be enough in translocations. So black rats, for example, we need to consider the control of those as well. And more broadly, the threat of black rats may exist in the wild for this species and for other species, and this has may have just been overlooked because we see such an impact from cats and foxes. There's been less attention paid to the effects of black rats. I also developed a low-cost monitoring technology that's also broadly applicable to a range of species. And I'm really grateful that I've been able to help already with the new one mouse captive breeding and translocation program in Victoria. So I've helped them with the development of some of their captive strategies as well as their translocation protocols, and especially that monitoring equipment as well, which they're using. So, yeah. Okay, so acknowledgements. I'd like to acknowledge first my supervisors, Stephen, Elise, Linda, Fred, and Marissa. You've all been very amazing. Thank you so much for supporting me and having faith in me and doing such like wild, crazy things. Funders and collaborators, so New South Wales DP, the ACT government, Holsworth Research Endowment, the faculty, Zoos Victoria, especially Phoebe Burns from that team, but the entire team. Melbourne University, Museums Victoria, Dr. Kevin Rowe, providing a lot of these samples and the sequence data. The Australasian Wildlife Group, Genomics Group in Sydney, at Sydney University, Gibson Water and Brooke Hay and Billy Lazenby for providing samples. I'd also like to thank the PhD cohort. You guys are all amazing. I couldn't have done it without you, and especially Emily, Matt, and Phil. And I'd also like to thank my partner, who has not only financially supported me through this, but for all my wild and crazy design ideas. He was there to help me make all of that tangible. So I couldn't have done it without you either. Thanks, Mick. The animal care team, who take great care of the animals. You guys are all amazing, especially Chelsea, Jackie, and Jamie, and Erin. Bronte Tozer, honor student who's not here today, but she played a big part in this as well. All the field volunteers who volunteered their time, so that was the animal care team, as well as people from the ACT government. And technical support, Bernd Gruber, who helped me a lot with all the dart stuff, and also like the organising of the sequencer. The EDNA team, and the faculty for general support, so Janine and Diane as well for all the support throughout this. So that's it. Thank you. Turns out doing this wasn't a good idea.
Did a brilliant job. Anyone got any questions? I'm sure there must be.
Yeah.
Oh, hang on.
Are you going to take out of mine?
I'll take the mic out.
Hey, Kristen. How did you select which individuals to release?
So it's a tricky one because we have like quite a small colony to start with. So ideally you want to select your fittest and best animals, but you also don't want to lose that trade-off by like losing the animals that you can't use in the breeding facility. So we had to kind of like balance that out. There was like big things when I saw negative, like really negative behavior. So any instances of aggression, when we did release, we released them separately as well. So yeah, typically you want to be doing like a checklist of all the behaviors and making sure they all have it. But the guys in the colony I've had were all pretty good. There was like maybe one or two individuals where I thought, okay, like you're not going to code. There was one example, Gus, who was like a colony favorite. And he's super friendly, and he has been since he's arrived from the other facility, and he will eat food out your hand. Like he's super friendly. And I didn't release him in the first round because I thought like you're just too friendly, you're not going to do well. I ended up pairing him with a female, and they didn't produce offspring, but he started to get all these wild behaviors back. So he was like really active, digging burrows, building nests, and he wasn't coming out to greet people, which was really great. So then I tried releasing him. And the day that I released him, I came back the following day, and he was like sitting inside the feed bucket in broad daylight, like just kind of eating. And I was like, that's not good because they're nocturnal. So he's meant to be in these burrows. I can't like shoot him out, put him back in the burrow. And then I went off, came back 10 minutes later, and he was perched up on a branch in the self-release pen, fast asleep in the sun. I was like, no, it's really good. So I scooped him up and took him back, and he's now back at the facility. So yeah, it's a thing of, and ideally you'd want to be managing for genetics as well. But again, we've got such a small, restricted kind of population and release pool that it's really like whatever we can get, as long as it's reasonable, we release them. So
Thank you.
Who was next? We've got a few down the front.
Probably don't have the mic. Online people need to.
True. Okay. Sounds good. So you've set the bar really high for animal husbandry, as I think we all know. Do you still see any stressors or variables that you're still tackling?
Yeah, I mean, like there's little things like enclosure changes is always a big one during the breeding season. Like I've arranged it so that they have a nesting space, which is a substrate that doesn't need to be clean. I like leave that for months on end because it's a really absorbent substrate. So we can leave that, but there is times where you have to go in there for various reasons. Like sometimes they bring food in there. Sometimes you just have messy animals that just pee everywhere and it stinks and you've got to clean it. So that always disrupts things a bit. I think, you know, apart from just not doing any husbandry routines, which is impossible, I couldn't really pick anything at the moment. It's all working really well. And having that bioactive substrate, that natural substrate helps because that's like kind of works on its own. I've got springtails in there and there's like things growing in there. I scatter seeds. So that requires very little cleaning as well. It's very much in the dot. So yeah, so that's a big help. But yeah, not that I can think of.
I just want to ask something before Arthur, it's quick. With collaborating, are you still seeing issues with, I find with endangered species, different groups and collaborators kind of hoard information over endangered species? Are you finding with captive now with the smoky mice, because you've got it so organized, that working with other teams has been easier? So for example, like Tib and Biller and using their facilities, do you see any problems in the future with that, or it's
I think generally I've been pretty lucky. There are cases where things haven't worked well, and there's been like a bit of secretiveness and nastiness going on between organizations. But But I think I've generally been pretty lucky and everyone's been very supportive and very interested. And because I'm really open to sharing, I think that helps that as well. If I come and I go, hey, I've got all this stuff, like if you want information, like I'm happy to share, like because I'm really open, I think it helps other organizations be quite open and communicative as well.
Yeah, very impressive body of work, great. So I just wanted to ask some questions around that disturbance. So disturbance is obviously it's sort of context dependent. So it's possible that if you just kept keeping them with a certain level of disturbance, that they gradually come to accommodate it. I guess the other side of to that is if you reduce the disturbance down, did you have any evidence that they became more sensitive to disturbance as a result of your reducing the disturbance? That's the first part of the question. The second part is, if they have accommodated the low levels of disturbance, when you let them go into the wild where there's another regime of disturbance, have you actually primed them for failure in terms of accommodating disturbance?
Okay, so first part of the question, you're asking whether
they became more sensitive to disturbance when you reduce the level of disturbance. So like that's an obvious thing that's likely to happen.
Oh, okay. So the first part you actually asked is like, do you think if you're continuing exposing them to disturbance, they'll get used to it? Was that
Yeah, and the other side of the coin is if
Yeah.
you reduce the disturbance, so they become more sensitive to disturbance.
So the first part, I think all animals will acclimate to conditions. When you're exposing them to things, they're going to get used to it. But they're very different to wild domestic animals. You don't like we're in there seeing these guys all the time. There are the young ones, the offspring that we handle quite regularly. They're not breeding. And they are still terrified of people, like going there all the time, you provide their food. They just don't have that inclination to warm to you like domestically bred animals do. They have a hardwired evolutionary biology to run away from everything that is bigger than them, anything that can potentially eat them, which is everything because they're a small bottom of the food chain. So I think that in there is like so strong. It's going to be really difficult and I think counterproductive to try and tease that out of them by just exposing them to conditions. I have found to an extent that they can be a little more sensitive to disturbances if you completely minimize everything, in the sense that so some of the animals we first got from the other facility were quite happy to greet their food. When you put their food out, they would come out, like they weren't so bothered by you being there. And now I find that if they have opportunity, they won't. It happens that they're out when you're in, they run back to the nest box. So I don't know whether it's those guys were used to it or they just didn't have opportunity. I think it's hard to kind of figure that out. But what I think is if you provide the opportunity to escape, an animal that's evolutionarily inclined to do that will and should do that. Yeah. Yep.
I was just wondering what your hesitation for recommending genetic rescue is. Do you think is there something you think needs to happen first before you would kind of go down that route?
Well, I think I'd like to talk to all parties involved in smoky mouse kind of conservation work and see where they sit on it. I know there are a lot of people that are very concerned about genetic rescue strategies because of the risks of the outbreeding. I don't see that as much of a concern. I think that's probably like a newer thing. I think people that are coming through now doing their PhDs recently are seeing the new literature and realizing there's probably not a lot of evidence for that being problematic. But I know there are a lot of people who are still concerned about that. And theoretically, I get the concern. So I think it's a balance of like hearing what everyone has to say, everyone's opinions on it, and just doing what's best for the species. And I think genetic rescue is required. Like I would recommend definitely consider it. If it was just me, I would go out and do it on my own. Like I'd like to have a group of people and hear those, weighing those opinions. But yeah, so that's just kind of where the reservation comes from, is that it's still not a clean cut, just chuck new animals and we're done. It does take a bit of consideration. And we do have like differences, a little bit of difference in physiology as well. Grampians mice are a lot bigger than the mice that we see out there. So, and I mean, could be related to genetics or not, but it's something that we, probably need to consider. Ben?
Yep. No, thanks. As Arthur said, impressive. I've got a question. Your captive breeding selection. You mentioned you have the principle to go only for lowest kind of relationship. Do you also think about there's some other strategies, like you want to breed different lines over time to keep diversity and not mix too much? Do you also use more involved strategies?
Yeah, so what I think has worked well, I mean, at the start of this, that the previous place maintained really good pedigrees because we didn't have genetic data yet. And what we actually found is that inbreeding, when we first started mixing, actually went down because these animals were selected from different sites, right? And when you started mixing them, you had this sudden mixing, and then so the inbreeding actually went down. And I think retaining separate lines for a period of time actually helps kind of delay that loss of alleles through that constant mixing. So I think as long as I mean, without doing kind of like a genetic analysis every season and looking at your form, I think it helps when you start with diversity to maintain lines for a while, and then you've got to figure out at what point you're going to start crossing, I think is the best strategy. So the kinship values, I mean, like that, we're at a point now where we started with 4 founder animals that have contributed to the colony we've got today, and that started in 2016. So it's a very limited gene pool. We maintain pedigrees at the start, and now it's a matter of like really just, you know, mixing whoever we can who's not brothers and sisters. So it's really useful from that perspective.
Can I very quickly, one more question, sorry, very related to that. We do the same for grasses earthquake, and we inform basically, hey, these should be the pairs, and then we are happily ignored because there are so many other considerations.
Yeah.
And is it the same there? So you find out this, that would be the best pair, but obviously they don't like each other, so it doesn't work.
Yeah, there is a lot else to consider in like a perfect, and I think actually I'm finding like part of it now that we've kind of standardized captive conditions, the overall reproductive success is a lot greater. So kind of we're having like more animals breed than they used to. So we can be a little bit pickier with things like inbreeding coefficients and genetics. Whereas before, like when you have issues like condition of animals and age of animals, like if you have premature mortality rates and you have this stochasticity going on, you've really got to put those factors first. So I think that kind of like those breeding pedigrees where you're really doing it based on genetics is more when you've got everything else is working really well and standardized. Everyone's happy, healthy and breeding, and you don't have a lot of variation in your stock, and that's very handy. But definitely there's a lot of other factors to consider.
Just have one question online.
How do I do that? Hello, Elise.
Yeah.
Hey, it's Michael Lynch. How you going? That was a really great and interesting talk, Kristen. And I'm a veterinarian, so of course I'm going to ask a question about health. And it's really interesting to see that direct predation of the black rats and the idea of releasing into predator-proof areas, but they're actually not predator-proof if the rats are there. But the question is really around disease, because black rats and smoky mice can share pathogens. And was there any sort of investigation of what was in the black rats before you release the mice, because they're sharing these same niches, and maybe even what was in the mice before they were released? And I guess you would maybe have some post-mortem data on that, like for the captive ones. And I'm presuming you didn't have the chance to recover any bodies or anything like that.
Yeah, so no bodies recovered. We do all the animals in the facility when they die, they go through post-mortems. So we are constantly testing for any disease risks and pathogens. So from what we know and what we've tested of, we haven't found anything in the captive colony. The black rats are definitely a risk for disease as well. We didn't really consider black rats that threat at all. Well, maybe very marginally, but they weren't really considered enough before these translocations started. I think like, you know, in hindsight, now that we're seeing what's going on, that they are a big problem, and there is a lot more of them in those areas than we thought they were. So when you actually start using supplementary feeding, they all come out of woodworks. So we didn't think they were going to be a big issue. Now that they are, that's definitely something that needs to be considered. So
yeah, I guess the thing is too that's the overlap of environments they're utilizing. So all those, you know, various pathogens that might be carried through, even fleas and things like that, they don't even have direct contact. So it'd be sort of interesting to know in the future, like what's in the rats in the site. That would inform your risk a bit.
Yeah, absolutely. And even for in the wild, you know, it's probably something that we need to look at in the wild where they're overlapping in their range as well.
Yep.
So yeah, good point. Thank you.
Great.
Yeah, I was just a question following up, I guess, a bit what Bern was talking about, and also the question of genetic rescue. And you did mention in your talk consideration about things like biobanking and assisted reproduction. Just wondering if moving forward, it's something you guys are considering implementing, because of course this can help a lot, provide sort of better control, rather than relying on your ideal pairs actually mating, because I guess sometimes they just choose not to and things like that. Being able to do genetic rescue, because you don't necessarily need to move physical animals that might harbour disease from population to population, and also just storing down what diversity you have in case there's inbreeding, depression and stuff like that as your breeding program goes on. And considering, you've got a fabulous mammalian reproductive biologist on your supervisory team in Marissa, and the experience down at Sue's Vic, is this something you guys are looking at?
Short answer, no, like we haven't discussed it seriously. Like I have been since you first come along and raised these questions, I've definitely been thinking about it, but it's not something that we've seriously discussed. And especially in light of like seeing how low the diversities in gene pools are and how high risk these wild populations are of extinction, I think it's definitely something that we should be looking at. Yeah.
Did you hear that? No.
Yeah, thanks. Really interesting. With that 250 year decline, where the population start to decline, how accurate is that estimate? And what I'm thinking is, okay, yes, you and assign that to Europeans, because Europeans, yes, came to Sydney in 1788, as we all know, but until Europeans were in the or at least in any numbers in the Grampians and Kosciuszko area, that was decades and decades later. So is that just a function of uncertainty in that estimate, or are you thinking something like the rats and the cats, etc., are running ahead of the Europeans and causing havoc before Europeans got there in any numbers, or what are you thinking?
So I don't think it's a direct at 250 years ago they started plummeting. What that is showing is a trend that around that time. So I think that's going to be a bit of give or take either way. So I think there probably would have been a delayed effect, and maybe what's happened is that they've started big dropping off so quickly, say in the last kind of like, maybe 50 to 100 years especially, that's that rate has declined so quickly that kind of trend we're seeing is just a product of like a really rapid more recent time. But it's because it's not, I wouldn't say at 250 years we suddenly started seeing it. It's really within, and most of those populations, there was a couple where you started seeing it 250 years, and then there were a couple where you were seeing it a little bit later as well. So it was like give or take, I think. Yeah, but yeah, I don't know about the rats and cats getting ahead of where anthropogenic impacts were. I can comment on that one. I'm sorry.
Maybe have time for one last question before we all go grab lunch. Anyone? Thought I saw a hand up over here somewhere. Oh, all right.
Okay, hopefully a quick one. Congratulations on the success of the mice at UC. I was wondering if you had plans to share the procedures or design of that with other teams who are working with smoky mice.
What, sorry, what was the last bit?
Yeah, with other teams who are working with enclosures of smoky mice. To be like, look, I got the gold standard. Go check us out.
Yeah, so there's actually only one other group at the moment working with smoky mice, and they have access to all this information as well. So I'm definitely happy to share, and with similar related species as well. So like I mentioned, the Newland mouse, which has been really great to be able to share with them. But I am wanting to put it together in like a really detailed step-by-step framework, which will be a publication. And I would like that to be applicable to really all species, especially small animals and especially small mammals. So, but yeah.
Awesome. I think we should let her off the hook. She's done an amazing job. Well done, Kristen.